In this video, we will be talking about Nirenberg and Mattaei's and Nirenberg leaders' approaches to genetic code. In the year 1961, Marshall W. Nirenberg and J. Heinrich Mattaei set out to determine which codons codes for a specific amino acid. They started with an experiment known as the poly-U tube experiment. Cell-free extract was used to emulate all cell components. The cell-free extract was mixed with a string of U's, aka poly-U, mRNA, and 20 amino acids. Out of the 20 amino acids, one of them was radioactively labeled. The tube was then incubated and translation took place. They did this step 20 times, each time with a different amino acid. The radioactive protein was coded for the original acid that was labeled radioactive, which was phenylalanine. Therefore, the U's encoded for phenylalanine. They repeated this in other experiments with poly-C, poly-A, and poly-G. Poly-C coded for proline, poly-A coded for lysine, but poly-G never synthesized any radioactive proteins. Mattaei and Nirenberg made great progresses in solving the genetic code. But Nirenberg continued his work with Leader to learn more about the genetic code. They set out to determine other matches between codons and amino acids with the use of tRNAs. Short mRNAs with known codons were synthesized and added to a mixture of ribosomes and tRNAs attached to amino acids. The ribosome bound the mRNA and the tRNA specified by mRNA. Then the mixture was passed through a nitrocellulose filter, and if binding occurred between tRNA, ribosome, and codon, then it would not filter through the paper, and the non-bind would get filtered through. This way, they were able to identify 61 codons. By the year 1966, Nirenberg and other scientists had translated all 64 RNA three-letter codes for all 20 amino acids, which we now know as the coding dictionary.